genetics and genetics in bacteria. And genetics in bacteria, well, all genetics really boils down to DNA. Okay, so this is one of my favorite cartoons showing that the structure of DNA was solved in about 1953, and I think a lot of people just think of DNA as this long, twisty, rope-like ladder thingy, but it really is the heart of all inheritance. So DNA is a nucleic acid, and the point of saying this is that nucleic acids are what's going to store and transmit all heredity information. Okay, and that doesn't matter whether you're in a prokaryote or a eukaryote. That's always going to be true. Okay. Now, the amino acid sequence of a polypeptide, so of a protein, is programmed by this thing that we call a gene. Okay, and genes are made up of DNA. That concept that genes are made up of DNA is really the heart of genetics. And while we knew before 1953 that there had to be some genetic material that was what was inherited, it just wasn't known what it was until 1953. There are two types of nucleic acids: deoxyribonucleic acid, DNA, and ribose nucleic acid. DNA provides all the instructions needed in order to replicate itself. DNA can direct the synthesis of messenger RNA, and that messenger RNA will later be converted into a protein. The protein synthesis, making the protein from that gene, occurs on the ribosomes. Okay. So just a little bit about nucleic acids. So here is one strand of the nucleic acid, and blown up here in the center. What's important here is that you have a sugar, a nitrogenous base, and some phosphate group. Okay, and this is all the base pairs, or every base pair is going to have that structure. In a typical bacteria, you might have 5-6 million base pairs. A human would have at least 10 times that. Okay. Now, the nitrogenous bases are there can be 4 at any time. So if we're in DNA, you have cytosine, thymine, adenine, and guanine. And if you were in RNA, you would have cytosine, uracil, adenine, and guanine. There are rules so that A always pairs with T and C always pairs with G. So when you're looking at a double-stranded DNA, you're always going to have these in pairs. Now, depending on whether you're in DNA or RNA, you have a slightly different sugar. Start here. The ribose has a hydroxyl group at 2 different positions, where in the DNA we're missing one of those hydroxyl groups at the 2. Now, one of the great things, or one of the just things that have happened, is that RNA lasts about, on the order of minutes, whereas DNA can be on the order of thousands of years. And we certainly know that we can isolate DNA from mummies in Egyptian tombs, and so almost, it's the DNA is so stable that it's essentially stays around forever. And one of the great things about that, I mean, look at, there's very little difference, right? The bases are basically all the same, and the only difference is this loss of this hydroxyl group. But being the no hydroxyl group at that 2' sugar is what gives us that increased stability. Again, just a close-up of the nitrogenous bases. These are in two groups, the pyrimidines and the purines. The pyrimidines are just single ring structures, whereas the purines are 2 rings. Okay, so we always have to fit it together so that we have a 1 ring and a 2 ring, or in this case with thymine and adenine, a 1 ring with a 2 ring. If you, for some reason, had two rings or two pyrimidines across from each other, it just wouldn't work. The DNA helix would just be the wrong size. And I've already covered the sugars with the DNA having just an H at the 2' position, and in ribose, the 2' position has an OH. And like I was saying before, this OH makes RNA much less stable. So up here is a picture of Watson and Crick in the model they first built to describe how the DNA was formed. Watson and Crick were the ones who won the Nobel Prize in 1953 for finding the structure of DNA. And here's a cartoon of the DNA being replicated. You can see up here at the top, this is the unsynthesized part, where you have these ribbons that are on the outside, and that's called the sugar phosphate backbone. So that phosphate group that I showed and that sugar are what's in these ribbons. Then the nitrogenous bases then are the in the pairs of CG and AT, and those are bound together with hydrogen bonds. Now, the reason that that's important is that hydrogen bonds are relatively weak bonds, and so we can very easily pull these two ribbons apart so that we can replicate it. So here you can see that the blue is the old strand, and then in the orange, these are the new strands. And so every piece of DNA always ends up being a copy of the old strand and the new strand. And the real thing that is important is that you always keep the base pairing, right? That you always have your A's and your T's and your C's and your G's. Now, this is kind of fun. These are a couple models. The majority of DNA that's going to be in a cell is in what's called the B form. And in the B form, you have this nice, gentle curve. You can see it a little bit better in the space-filling model, where you have a right-handed helix that, this is the form that is mostly in cells. Now, there is a second form, the Z form, that can be in cells. It's very seldom seen in microbes, but there are eukaryotes that do have this Z form, and the Z form is basically twisted backwards, and instead of being a nice gentle curve, you get this kind of zigzag pattern. And one of the great things about being able to sequence DNA is that we can use that information to explore evolution and evolutionary kinships. So by knowing what the sequence of DNA is, you can find all this information. So depending on how similar two different DNA sequences are, or how closely related a certain gene is, you can see how they have evolved from each other.